Okay. Alright, if you remember from the very, very beginning, we talked about those 4 macromolecules, right? It was carbs, lipids, proteins, and nucleic acids. DNA is a nucleic acid, so let's put out of the macromolecules, nucleic acids include DNA and RNA. Can y'all see that?
Can I move this?
Yeah. When we talked about the macromolecules, we said that each of these macros has a monomer, or the smaller units that make it up. So, nucleotides are the monomers of nucleic acids. And the nucleotides have 3 parts. If I get to going too fast, y'all make me stop, slow down. The nucleotide has 3 parts. It has a phosphate. What's a T? The sugar, deoxyribose, and a nitrogenous base. Now, before I've always referred to this as the pool, the house, and the garage. So the pool, the phosphate, starts with a P. The house is where you keep your sweets, like the stuff that you eat. And then it has like this little garage over on the side here. So like we've added up here already, the nucleotide is the monomer of a nucleic acid. Within these nucleotides, there are 4 different nitrogen bases. They are adenine and thymine, cytosine, and guanine, and I did them in matching colors because those are going to always pair up together. And then we've already added up here too, the sugar found in DNA is called deoxyribose. Alright, I'm going to scoot over on this side. So DNA, we haven't really mentioned it yet. DNA stands for deoxyribonucleic acid. So deoxyribo is the sugar. It is a nucleic acid. So deoxyribonucleic acid is DNA. Alright. We're going to label the 4 bases. Let's do them all the same. So they all have the little pool, the phosphate. They all have the same sugar, the deoxyribose. I'm going to abbreviate, we don't have room for the whole name. Alright. There are 4 different base options. So you have adenine and thymine, which are complementary base pairs. Adenine and thymine are always going to pair together in DNA. Cytosine and guanine are always going to pair together in DNA. So think for adenine and thymine, apples in the tree. Cytosine and guanine, cars in the garage. Guanine. This one?
No.
This one?
The very first one.
Oh, adenine.
No, it's not the bottom. No, in the parentheses.
Oh, complementary base pairs. Okay, let's flip. Well, y'all can't flip yours over. Let's see, where can we put? Let's draw a picture of DNA up here. If DNA were straightened out, it would look kind of like a ladder. Each side of this ladder, kind of like you'll see in your little worksheet, is going to be a different nucleotide joined together in the middle by these complementary base pairs. Because DNA is so long, it coils up and condenses into those chromatids or chromosomes, right? So that's what these are made of, is DNA. And because of that, it is generally seen in this like twisted shape. So if you took your ladder and you twisted it, this is what you would see for DNA. Let's practice matching some, and then we'll move on. So I'm going to list some DNA bases, and we're going to find the complements. So let's say we have ACG, TAA, CCC, AGT. I have clustered these in groups of 3. These are called triplets. Later, when we look at RNA, this is going to be really important, but we read in sets of 3. Now, the complement is going to be whichever one they pair with, however we color-coded. So what goes with A? T. What goes with C? G. What goes with G? C. What's next? What pairs with T? A. What pairs with A? T. So everything has a match. Alright, makes sense? So far?